How's everybody doing? What was that?
Good.
Good. You guys glad it's Friday?
Very.
All right. Well, good. You guys have any big plans for the weekend?
Testing, can you guys hear me okay?
We can.
Okay, good.
No trick or treating or anything this weekend?
1 second.
Yep. I actually plan to come to your house and maybe get some candy. I brought a new basket and everything. So
Actually, I'm not going to be there tomorrow night. So I'm going to be at my girlfriend's mother's house. So our lights will be off, but our pumpkins will be lit because they're electronically lit. There are no candles in them. We thought about leaving a bucket of candy on the porch, but then we figured the first two people to get there would just unload all the candy.
That's weird.
So we decided against that ultimately.
I can't
So
hear you. And
is there anything on?
why?
Do you need your headphones on, Nathan? Of course, he can't hear me, which is the reason of the problem. Is there anything happening on campus this weekend? Halloween related?
I'm not sure, really.
Like stand 6 feet apart and throw candy at one another or anything?
That sounds kind of fun.
Depends on the candy. Reese's peanut butter pumpkins, that's not so bad. Jolly Ranchers, those could hurt depending on how fast they're coming at you.
I think it's funny that they have the Christmas and the Halloween decorations up right now.
Oh my gosh. I walked into Lowe's at the beginning of October and they were moving aside the Halloween stuff to make room for the Christmas stuff. And I'm like, that's crazy. That's why it snowed before October, before Halloween. All these people wishing winter to arrive early. I'm pretty sure that's how global air currents work to bring winter on early. I'm cracking Dr. Judd up because it's geeky science humor. Dr. Judd, do we have any announcements or anything?
I don't think I do. We read our thing from yesterday.
All right, cool.
Yeah, I just had a little technical issue, but I'm ready to go. So we're going to finish up our conversation about measuring biological diversity, and then we'll transition via some prior organisms of the day into our discussion of photosynthesis. Right. So last time we sort of went through a little bit of your preparatory homework, and I hope define some terms, define things like species richness, species diversity, and species dominance, and how those are all interrelated. We also talked a little bit about species composition, which has to do with not just how many species there are or how common they are, but their identity. And that is what is expressed in a species accumulation curve. So as you encounter new individuals, you have the opportunity to encounter new species because individuals belong to species. As you add species up over time, you accumulate species. So those things can all be, well, at least the first three, can all be pretty well summarized graphically with a rank abundance diagram, also sometimes called a Whittaker plot. I say Whittaker plot because there are several different ways that you can plot species diversity and abundance, or species richness and abundance on a plot, and 3 or 4 different ways show up in literature. But this is the one that we want you guys to be familiar with. As you can see, it's pretty easy to figure out the number of species in a community or a sample of a community. It's pretty easy to get a sense of how even or the relative evenness if you compare two communities, like we did last time with this one showing a uneven community with high dominance, community 1, and a rather even community, community 2. These are pretty extreme in either direction. And we ended with these questions about what might influence richness and evenness. But now what I want to do is talk about these metrics, these indices or metrics that we use to summarize the information that you can see visually within a rank abundance diagram. And we use these metrics because it allows you to get a single, or in this case, 3 numeric summaries of the information that you can see pretty easily on the diagram. And then you can compare many different communities all at once, or compare many samples from within different communities all at once, as is done in the Wien et al. Paper. So we're going to talk about the Shannon Diversity Index, Simpson's Diversity Index, and Pilleu's J, which is also called evenness. And you'll have some opportunities, or you'll want to practice doing these calculations by hand, and then maybe using Excel to check your work. But we'll talk about the mechanics of how we do these things and what's different about each one of them, and why you'll often see ecologists use 2 or 3 of these at a time and not just one, ignoring the other 2. So the Shannon Diversity Index, usually denoted by H prime, this little H with the apostrophe after it. This comes from the field of linguistics, actually, and code breaking. It's kind of an information content metric, but we're going to think about it in terms of species. So what you need to know about this is you can think of it as if individuals were randomly associated with a species and the species had varying abundances, you would kind of imagine a community like a jar of marbles with different colors. And the question is, if you reach into a jar of marbles where there's 10 different colors and they're all equally abundant, then you're going to have maybe a 1 in 10 chance of guessing correctly the color of a marble that you pull out. If your community is very uneven, such that 90% of the marbles in your jar are one color, and then there's 9 or 10 marbles that are various other colors, you can be pretty confident that you'll know if the red one is the common one, that's the one you'll pull out. That's kind of like what this H value, the Shannon Diversity Index, does. And what you do to calculate the H value for your community is you have to figure out this P sub I. It tells you the proportional abundance of the ith species in your community. So remember, I stands for index. So you index through all the species in your community and you obtain a proportion or an abundance metric for each one of those. Then you take the natural log of the abundance of a species, as this ln parentheses r, and you multiply that by the proportional abundance again. So it's pi times the natural log of pi. And you have to do that through all I of your species in the community, whatever number I is. It could be 10, it could be 20, however many species are in your community. And then you sum all of these together, and then we multiply it by -1, so you get a positive number, because when you do this, you'll get negatives, so we make it positive, and that's your H prime. This metric is kind of entrenched in the literature, and you might think, oh, well, you could have 2 different opinions about it. You could say this is a really good metric because it summarizes both the number of species in the community and their relative abundances. Or you might think this kind of confounds the abundance of different species and how many there are. You could have 2 communities with different richness values, but similar or nearly equal H values if their abundances are different in such a way as to get you the same value. So it has some issues that some people see as an advantage and some people see as a disadvantage, because it summarizes a lot of information. It assumes that all individuals are sampled randomly from your community, which is maybe not a perfect assumption, but usually it works. And usually the metric is robust to that. And it assumes that all the species in a community are represented in your sample. If you have unsampled rare species, then the H value is going to miss those. They're not going to be in your P sub I data set. One thing to know about this metric is that if you take E, the constant E, and you raise it to H prime, so I don't have if I had a whiteboard here, I do have a whiteboard right behind me. Stop share for a moment. So say you calculated an H prime value for your community of 2.35, which is not unusual. It's usually somewhere between 1 and 4. If you were to calculate E raised to the 2.35, then that would basically give you the number of species that would have been found in your community if it was perfectly even, if each species had equal abundance. So, and that has the effect of spreading out the numbers. Remember, I just told you that it usually varies between about, you'll pretty much always get a number between 1 and 4 for H prime, which is a pretty narrow range. If you use this E to the 2.35, it kind of spreads out the data and gives you kind of a different metric. But this is traditionally what people use, the H value. And it was developed by Claude Shannon, who is a linguist, as I recall, who's shown here in the lower right on the screen, which I will share again. So that's one of the 3 metrics that you're going to see. The next one is a measure of evenness. And sometimes it's just called evenness, and sometimes it's named for the woman who developed it, the ecologist who developed it, Evelyn. I forget her first name actually now, I'm blanking on it, but her last name is Pilot. She's shown there in the lower right. And she knew that if you raise H prime to, or if you raise E to H prime, you'll kind of get an understanding of evenness in the community. And so she developed kind of maybe a more intuitive way of doing that, where we divide H prime by the natural log of the species richness. So if you divide H prime by the natural log of species richness, then you get this measure of evenness. And the natural log of S is basically the maximum H diversity that you could get if it had been a perfectly even community. So I don't actually recall offhand, I could look here in a moment, if they use J, but this is the metric of evenness that is used in the paper. So know how this is calculated and how it relates to H prime. And the last one is the Simpson's index. This one is more of a metric. It's usually framed as a metric of dominance. So a high D value is a community that's going to be very uneven. But sometimes it's recalculated as an index of diversity by just doing 1 minus D. So notice the D ranges from 0 to 1. It's going to be a decimal no matter what. And then if you flip it around 1 minus D, then you get this index of diversity. In this case, we have a capital N, which is the total number of individuals in your community. And the lowercase n is the number of individuals in the ith species in your community. So n sub I for your first species, n sub 1, times n sub 1 minus 1. So you've got 20 individuals. That's going to be 20 times 19. And then that's going to be divided by the total number of individuals in the community times the total minus 1. And you'll index through all I of your species in your community and then take the sum of each of those. That gives you this D value, Simpson's D, which can be flipped around to be index of diversity. Once again, it's a fairly simple equation because it makes certain assumptions that are usually fine to make in a study like this, where the individuals are randomly sampled and the samples are independent of each other, those kinds of things. What does all this look like? What does applying these things look like? Well, you might have a plant community and you've collected your data and you've found there are, several species. You've figured out what the identity of each of those are. Notice we have a grass that's rather abundant. We've got another grass that's rather abundant. A bunch of these are grasses. Baptisia is a legume. Asclepias is a milkweed. So you've got some common prairie plants that are abundant, and then several rarer things that include both grasses and herbs. You count up the number of individuals, and then you calculate the proportional abundance of each of those. Then in the next column, you can take the natural log of that, and notice it becomes negative. Multiply those together and take the sum, and you get that P times ln P. And when you add them all together, that gives you your H prime. You can also easily take these numbers and calculate Simpson's index using the n times n minus 1 and also calculating it over the product of the total number of individuals times that number minus 1. And notice the relationship between D, Simpson's D, and the Simpson's diversity index, right? If you add those together, it's 1 because the diversity index is 1 minus D. Any questions about how you could, about these three indices and how they relate? And do you feel like you could create a table like this in Excel if you had to?
Can you explain again what the variables of Shannon's diversity stand for?
Yeah. Let's go back to. So natural log, right, is an operation. And the this is the sum. This is the indicator for the operation of take the sum of in math. So all you need to know is PI. It's the proportional abundance of each species in your community, and the number of species in your community depends on the community you're sampling, right? So any given community will have I species in it, some number. In the example I just showed, I think we had 12. I'll go back to that in a moment. So you need to know the proportional abundance of each of your species in the community. Come on. There we go. So PI is the proportional abundance of, in this case, Poa pratensis. 0.1026 is 12 divided by 117. Right? 0.094 is 11 divided by 117. Make sense?
Yeah, so the 117, is that just all of it added?
Yep, the total number of individuals in the community.
Okay, thank you.
Yeah. Okay, so at Konza Prairie, the way they sample diversity within a watershed is to set up these transects within a watershed. These little lines here indicate the transects. And then along those transects are plots, and within the plots, they sample all of the diversity. And remember, they had some areas within the watersheds that were grazed and some that were ungrazed. So here's an example of some data that they collected to compare the richness of the plant community with bison and without bison. Can someone remind me what cumulative means? We've got cumulative species richness in an area with bison present and an area with bison absent. What do we mean by cumulative?
Like all over the total of the species.
Yeah, the total number of species, where we're accumulating species or increasing the number of species encountered, but we're doing it in a way that accounts for the identity of those species. So if one patch has 14, one plot has 14 species in it, and the next plot has another 14 species, are you at 28, or do you have some repeats? If you have some repeats, the total number of species is going to be less than 28. It's going to be maybe 16 or 18. So notice that as the number of plots increases, the number of species increases, but eventually you get to a point where you're still encountering plants, but they're not belonging to new species that you've never seen before. So let's look at this graphically. It's a little easier to understand. Number of plots sampled along the x-axis and species richness, cumulative species richness along the y-axis. So let me ask you, what's the effect of bison activity on the plant community, on species richness? Someone else, let's see. Is there a gallery view?
Bison activity increases species richness.
Yeah, good, thanks, Keaton. It seems that species richness is higher where bison are present, right? Who wants to hypothesize as to why that might be? Someone else. Grant, what do you think? Do you have any insights onto why bison activity might increase species richness?
So like the way I see it, like there's some like activities bison can like do that might, so like say like feces and like that can put nutrients in the ground, and say there's a species that thrives on that sort of nutrients. So like bison being there will help those species to thrive, which will increase the species richness.
Good, yeah. We're thinking about heterogeneity, and bison activity on the landscape is very local, whether you're talking about a pile of feces or a wallow or something else they do. That highly local disturbance or changes to the environment increases the heterogeneity such that you're likely to encounter more species, more different species in a given area when bison are introducing that heterogeneity and altering the structure of the community. So we can also look at that same pattern with some of these metrics of diversity. Notice that we've got this H prime value, bison present versus bison absent. Which one's higher? Yeah, higher with bison present. Good. Which community or which treatment, I guess, bison present versus bison absent, has a higher dominance score, where you might find patches of land or patches of ground where one or a few species of plants comprises more of the biomass and more of the individuals than the other?
Bison absent, right?
Yes, so we got a higher value here for bison absent, higher dominance. Good, and we will talk about why in just a moment. So fire is another source of heterogeneity besides bison. And as Dr. Kowinski was telling you in a previous lecture, was that the bison like to go to the recently burned areas where the new grass is coming up. The grass that tends to come up first and take advantage of those bright light conditions is nutritious, is easy to digest. The bison prefer that stuff. So they'll come in, and what would initially be a very low diversity, high dominance plant community is then manipulated by the bison because they're eating that common grass that comes up first and giving space and allowing sort of niche space for other species to come in. So the community ends up being a little bit more diverse in the end than it would have been had the bison not been there to suppress the most common, most abundant plant species. So make sure you can think about the relationship between richness, evenness, dominance, and how they're all kind of encapsulated together in that, that diversity and mix H that summarizes everything. Okay. So we have scheduled 2 days to talk about photosynthesis. And I went a little bit over last time with measuring diversity that we just finished up. So I'm going to try to be careful with how I use my time on photosynthesis. We might bleed over a little bit. So we're going to do a short prairie organisms of the day. First one I've got for you is our prairie forb. This is Salvia pitcheri. This is one that we'll talk about a little bit soon. It's a member of the mint family, which is a good family to know. It's pretty easy to recognize the mint family because they usually have aromatic leaves, which this one does too. If you crumple leaves up and smell them, it smells, well, fragrant. There's a lot of different scents they might take, but they almost all have a fragrant leaves. They often have square stems. So if you grab the stem of a member of the mint family and you kind of roll it around in your fingers, between your fingers, you can feel the 4 square sides of the stem. That makes it really easy to identify. This is in the genus Salvia. It's really a pretty one with blue flowers on these big spikes. Notice, anyone recognize the grass in the background here? The little turkey foot inflorescence.
Big blue stem.
Yeah, nice. So we've got it growing here with big blue stem in a prairie in southern Illinois. It flowers from July to October. I think some of you in my lab, we saw this out at the sanctuary. It's known for having really deep roots and being pretty tolerant of dry conditions for a forb. And root depth increases with drought. So in drier climates, you tend to find deep roots in these things. All members of the Salvia, genus Salvia have a variation of a really interesting and fun pollination mechanism. So if you look at a salvia plant up close, it's got this big lobed petal that's kind of a landing pad for bumblebees and butterflies. And then it's got a tubular corolla, this little blue tube that goes down here and is surrounded by the calyx. Down in there is where the nectar is that the bees will go for. Now, all members of salvia have this upper lobe of petals, and tucked in there are the anthers and the stigma. And the anthers are modified in a way that it's kind of like a lever. And this is a generic diagram, not of Salvia pitcheri, but it kind of shows how things work. There's a ovary down here with a nectary around it that produces the sugar water produces the nectar for the visitor. It's got a style here and then a little 2-lobed stigma, kind of like the one we saw with the asters. But then the stamens are, the filament is elaborated in a way that it makes kind of a lever. so that whenever the pollinator goes in for the nectar, whether it's a bee or a butterfly or a hummingbird, depending on the species, it touches one side of the anther called the connective, and the pollen producing portion then tips down and taps the bird on the head or taps the bee on the back of the thorax and deposits the pollen in a very special spot. So the example would be a stamen that's shaped kind of like my hand. The visitor comes in. To get to the nectar, it moves the bottom lever, and the top one comes down and taps it on the head. So that gives it a very precise placement of the pollen whenever it goes and visits another flower. And the stigma then comes down and touches the pollinator on the back of the head or the back of the thorax and picks that pollen up. And so there's ongoing work about whether this high specificity of pollen position among salvias has fueled adaptive radiation, where different species put the pollen in different spots or different parts of the body of the pollinators. So the lever system is kind of fun, and you can, if you find a salvia flower, you can activate it and see the little yellow pollen come down. All right. Our prairie grass of the day, I've got Panicum fugatum. This is switchgrass. This is a grass that's getting a lot of attention now because it's used as a biocrop. People grow this for ethanol. It's a common grass that's forming, got these big open panicles that are really distinctive, that kind of almost like a lacy look. It's really spreading, usually kind of reddish, kind of a loose sod former, and an important part of tall grass prairies, not just for its ethanol crop, but also it has some interesting relationships with other organisms. There's a moth that specializes on just eating its larvae just eat the foliage of this grass. And it's one of the more common or more dominant grasses that you might find in a tall grass prairie or a mixed grass prairie. And then I picked the 5-spotted hawk moth, another insect. Paul, maybe you like to do this one when I stole it from you, but it's a fun one because the larvae, the caterpillars of these hawk moths, are called the tomato hornworm, and it's a known pest of members of the nightshade family, like tomatoes and potatoes.
Nathan,
But-
I'm not seeing your screen anymore.
Oh, thank you. Let me just go back one slide so you guys can see the panicum. Here's that open inflorescence of the grass panicum with its leaves and the moth that specializes on this grass. Okay, 5-spotted hawk moth. So here you've got a moth, adult moth, pollinating what is probably a datura. It's a white flower. Many moth pollinated flowers are white. That's something you can usually, kind of a rule of thumb you can go with. Member of the nightshade family, the plant is, which is the same family that tomatoes and potatoes belong to. The larvae, or the caterpillar, if you want to call it that, the larvae of the hawk moth is called the tomato hornworm. Notice the bright green coloration and the white stripes and this pigmented, what do you call that, Paul, on the end? Is it the horn?
It's the horn.
Yeah.
I think that's
the
just what it's called.
Yeah. And this is a known pest of tomatoes and potatoes and things like that. But there are parasitic wasps that attack this this caterpillar, and so farmers and anyone who's battling this this hornworm as a pest might encourage the parasitoid wasps to infect the hornworm. They live inside the body, kind of eating the internal organs of the hornworm. And then when they are ready to pupate, they emerge through they emerge outside of the hornworm, and each of these little pupae here, from each one will emerge a new parasitoid wasp, which will go around and look for other hornworms, lay the egg inside, and then a whole population or a whole litter of these parasitoid wasp will grow inside the hornworm as it eats. So it really diminishes their ability to grow, and they obviously don't metamorphose into the moths, and the wasps do quite well, taking advantage of the hornworms.
For just one thing you might also want to add to this, you notice how long the proboscis is on that hawk moth? The mouth part enables the hawk moth to basically access the nectary of that flower, the place where the little sugar water is, that flower produces to attract them to the flower, and it gives them this food reward. But presumably that food reward is a food reward for coming and picking up pollen. But you see that that moth is nowhere near the anthers of that flower. So when you guys are thinking about pollen load for the pollen load group in lab, and when you're thinking about pollen limitation, this should get you thinking about what are some strategies that insects have to avoid becoming kind of roped into the service of flowers for pollination services. That long proboscis keeps that moth away from the anther so that they don't pick up any pollen. And so that moth is getting nectar without providing any services.
As I recall, sometimes this situation that you see with the moth here that Paul pointed out doesn't necessarily evolve because there's a cost to the moth incurred by carrying pollen around. Sometimes there are predators like spiders that will live on flowers and await for pollinators to come visit. Maybe you saw some crab spiders waiting for pollinators to come visit on the goldenrods. And so this is a strategy to access that resource, but not be vulnerable to the spider predator. But it has the consequence of the moth is no longer an effective pollinator of the flower, but it's stealing that nectar reward.
And there are some flowers that have evolved really long nectary tubes in order to draw the moth further into the flower as
so
well.
It does effectively pollinate. Yeah.
What's that?
So that it does effectively pollinate by being drawn.
Yep.
Nice. Thanks for that. That's a good reminder. That's a good one.
I'm just trying to pull some things into lab so
Yeah.
it integrates a little more.
Okay. Photosynthesis. This is our next sort of unit in the VNet All subunit, or the Kanza Prairie subunit. And what I want you to be able to do is know the inputs and the outputs of photosynthesis and the two parts. We're going to talk about sort of two phases of photosynthesis, the light reactions and the dark reactions. So I want you to know the relationships between the light reactions and the dark reactions, and the relationships between light availability and carbon dioxide as they relate to photosynthetic productivity, to the production of sugars by plants. We're going to talk about two different kinds of photosynthesis that we've already sort of hinted at several times with the prairie organisms of the day, C3 and C4 photosynthesis. I want you to know how those two are related and under what conditions the environment might favor one or the other. So that's kind of what we're getting at here at the last one. Predict whether C3 or C4 species will have higher productivity under different environmental conditions. And you should be able to explain why that is for any given set of conditions. So most of you know that plants photosynthesize. I want you to know that plants also respire. And the relationship between these two reactions is what drives much of life on Earth. We've got the process by which light energy is used to oxidize water, to obtain those electrons and those hydrogen ions, to make energy that fixes carbon dioxide into sugars. So this C6H12O6, this is our sugar. Atmospheric or gaseous oxygen is our waste product from photosynthesis, and the inputs are carbon dioxide, water, and light as the energy to drive this reaction to the right. You can think of respiration as sort of the opposite. It's the process by which these complex organic molecules like sugars are oxidized, broken down, to release chemical energy or to make chemical energy available. And the waste products are water, carbon dioxide, and heat. And this 36 ATP, that's the chemical energy that's made available. You can think of ATP as the energy carrying molecule of the cell. And by oxidizing sugars, chemical energy is made available in the form of ATP. Now, both of these reactions oversimplify a lot of stuff that's going on. And we're going to break down some of those things after a little thought experiment. So our thought experiment is going to be with radish seedlings. So imagine you've got 3 batches of radish seeds. You've weighed each of these batches of seeds so that each of them are 1.5 grams. They're going to have about the same number of seeds in each treatment. Then you're going to put your radish seeds into these 3 different treatments. One of them is going to be seeds placed on dry paper towels in the light. The second one is seeds placed on wet paper towels, also in the light. And the third one is wet paper towels, but this time we're manipulating the light environment such that they're in the dark. You know the initial weight, the starting weight of your batch of seeds, and you know these 3 conditions. After 1 week, you're going to take all the plant material that's still there, dry it in an oven, just like we did with the leaves and stems of the goldenrods, until there's no water left, and then measure the plant biomass in grams to see what's changed and why. So what I want you all to do is in your notes or in the chat, I want you to predict, let's do it in the chat, predict the biomass for each treatment in these 3 conditions. So 1, 2, and 3, predict a biomass for each one. It could stay the same, it could go up, could go down, could be whatever you think your hypothesis is. What's going to change given these 3 treatment conditions? And I'm going to watch the chat as you make your predictions. One week after you set it all up, what's going to be the change in mass of your radish seedlings over time, over 1 week? Same, so we got a couple of them are starting to come in. Keaton, what do you mean by same? Do you mean the same for as 1.5 or the same value for each of the 3 treatments different from 1.5?
Yeah, sorry, I was trying to make a list, but it accidentally sent. But for the first one, I meant it wouldn't really change much over the week with no water.
Okay. Increase, no change, little increase. Okay, good. Keep them coming. Nice, here we go. And what's our participant list today? 5. Nice. I like how we're getting a mix of qualitative and quantitative predictions. That's good. Just another moment. I think we're good. Almost everybody's in now. Okay, so I'm going to go to the next slide and show you all what happened. So we started with 1.5 grams. We ended up with 1.46, 1.63 when there was light and water, and 1.2 when there was water but no light. I wonder, there's a few of you here. I wonder, Ryan, let's say, what made you suggest decrease for the first one? Maybe I should have asked this first, but.
Because it's not getting anything it needs to grow.
So, and why, so why would you not say no change then?
Because it's drying out.
But we dried it out ourselves, right? Remember the,
Right.
you're not incorrect. I was just wondering if you could explain your reasoning a little bit. Let's see, someone else. Hey, Kat, can you tell me why you said decrease for the first one?
Kind of the same reason Ryan said, because there's no change. Well, now that I think about it, there would be no change, but I had the same thought process as Ryan.
And Cody?
I kind of thought the same thing as them too, for being honest.
So I want to remind you all, I'm going to come back to this slide. Those are good, reasonable answers. But I remind you all that plants photosynthesize, but they also respire. And what's the waste product of respiration here, indicated with the mouse?
CO2.
And where does that carbon come from? It comes from carbohydrates or sugar molecules in the biomass of the organism. So compare this 1.46 over here, especially where they actually do get water, even if there's no light. The mass decreased even more. Why do you guys think this went down in the case of number 3 even more than the case of number 1? What's different about seeds that weren't given any water and seeds that were given water? What happens to them? Actually, this is kind of a good question. When seeds germinate, when the seedling emerges, where does the energy for the seed to put up the first pair of leaves come from? If energy comes from the sun and they need leaves to get it, how do you get your first set of leaves going?
From like the sugar nutrients.
There's stored sugar, usually as starch, in the seeds, right? And if you're a seed and you're dormant, you only need to use a little bit of that sugar through respiration to stay alive and wait for those good conditions. So you'll have very small change, but it'll be a decline in total biomass without water, just enough to stay alive. But once you add water and the seed germinates, it's converting a lot of that starch into new plant tissue, into the little, the first root, and then into the hypocotyl, that little white part there of the bean shoot, that's called in this case, the radish, that white part's called the hypocotyl. And then the first 2 leaves to come out. Making all of that requires consuming some of that energy through respiration. And then in the case where you have light and water, what happened to the total mass of the seeds? Or the plants, I should say.
They increased and grew
And why did they increase? How come they were the ones that could?
because they had every component that they needed in order to go through photosynthesis.
And tell me about the balance between photosynthesis and respiration, given what you saw happened in scenario 3.
So if they can like go through the process of photosynthesis, they can also go through the process of respiration, but they're not emitting more than they're receiving, if that makes sense.
Yeah, I see what you're saying. And they're basically the sugar, the ability for them to produce biomass, right? Produce biomass using these sugar molecules is greater than the amount of sugar that they use up to grow as big as they did, right? So they're adding biomass because the balance is in favor of photosynthesis. There was enough light to drive this reaction strongly to the right faster than respiration. One of the reasons that plants are our photosynthesizers and algae are our photosynthesizers is that they don't have to do a whole lot, right? They stay put. They don't expend nearly as much energy respiring as we do as mobile organisms that have to maintain a constant body temperature. And because they are able to be sort of economical with their respiration, since they don't move, they can build up this excess, they can draw carbon dioxide out of the atmosphere, make sugar, and grow larger and larger. So the balance is in favor of photosynthesis when there's enough light, when there's enough water. And this should, in some ways, kind of amaze you, and sort of an easy thing to amaze somebody with. If you have the trunk of a tree, right, and you dry it out, you got a big log, and how many of you ever had to split firewood or something? Most of that weight that is the log before it was a tree was just carbon dioxide in the air. That's where the biomass, the dry weight, the carbon that is now the tree comes from. So how do you get carbon out of the atmosphere and turn it into a plant, turn it into a grain of rice, a tree trunk, or any other plant part? Well, that is the process of photosynthesis. This is a schematic diagram that I would normally draw on a whiteboard for you. And I want you guys to really pay attention to this diagram and know how it works. The little kind of bean shape here represents a single chloroplast. Chloroplast is an organelle inside the cells of plants and algae. It has a distinctive structure. It has these membranes inside, these lipid membranes inside the chloroplast that are called thylakoid membranes. And they come in these stacks, shown here in green, that are called grana, because they looked like little grains of some of the earliest microscopes that we used to see them. And it's in these stacked thylakoid membranes where part 1 of photosynthesis happens. We call that the light reactions. And this is the situation where light energy is captured and converted into chemical energy in the form of ATP and NADPH. In order to do this conversion, you need water as a source of electrons and hydrogen ions, and oxygen is given off as a waste product. As these energy storing molecules, ATP and NADPH, are produced, that energy is used to power the Calvin cycle. And the Calvin cycle is the set of reactions, a long, complex set of reactions that we'll talk about in a simplified way, that captures carbon dioxide from the atmosphere, fixes it to other carbon molecules such that it's water soluble, and ultimately groups those carbon atoms together as sugar or starch, expending this ATP and NADPH energy, and then giving those think of them as kind of spent batteries back over to the light reactions so they can recharge them and recreate ATP and NADPH. So we've got our energy production center and our carbon fixation reactions. Carbon fixation in this case doesn't mean carbon dioxide is broken. it means we're fixing it from a gaseous state or a dispersed state into a water-soluble state like carbohydrates. That's what we mean by fixation. Here's the same thing shown as a chemical equation. We've got our light reactions and our carbon fixation reactions. In the case of light reactions, electrons are obtained from water, breaking the water molecule, oxidizing the water molecule, and making those electrons are then used to create ATP and NADPH molecules. And then over here in the carbon fixation reaction, there's a protein, an enzyme called rubisco. Ribulose bisphosphate carboxylase oxygenase is what it stands for. Ribulose bisphosphate carboxylase oxygenase, but we just call it rubisco. That's probably the most abundant protein on Earth because everything green has a lot of it in its photosynthetic cells. It's a protein that reacts with carbon dioxide and sometimes with oxygen to make sugars, basically. So where does this all happen? Well, here's our little example plant. Here's a dandelion. Here's the leaf of the dandelion. If you were to look inside the thickness of a leaf, you would see an upper epidermis, a lower epidermis, and then a mesophyll, a middle leaf, mesophyll, middle leaf portion where many of the cells in here have chloroplasts, little green organelles inside the cells. If you look closely at one of these cells, especially in the upper layer just below the epidermis, you'll usually find a large vacuole, a storage organ for sugars and lipids and things. You'll find chloroplasts, and there will also be mitochondria mixed in here as well. So I picked a different image to show you a little more closely what's going on. This image on the right is a photograph or transmission electron micrograph of an individual plant cell. This thing on the right here is the nucleus. This white mass down here is the vacuole. These dark, long things, those are the chloroplasts, and the little dark ovals inside them are the grana and the thylakoid membranes. These smaller ovals are the mitochondria. So this is where respiration is happening, and this is where photosynthesis is happening. Chloroplast versus the mitochondria. And then we've got a drawing based on the micrograph over here, showing the same structure with everything labeled. You can see starch grains, you can see thylakoid membrane, indicated in green. If you take one of these chloroplasts and you kind of make it into a more easy to understand cartoon, this is what it looks like. Chloroplasts have a double membrane around them, and that reflects their history as once probably as being descendant from free-living, probably cyanobacteria. So they're kind of like a little remnant of a cell within a cell. You get a double membrane. And then inside the double membrane wall is the thylakoid membrane arranged in these lamella, and then sometimes stacked into these grana, or a single would be a granum. That's the thylakoid stack. So now we looked at a whole leaf, then we looked at the cells in the leaf, and then we looked at the chloroplasts in the cells, and now we're looking at the grana in the chloroplasts. And now we're going to zoom in once more and look just at the membranes of a segment of a granum. This is the structure of the granum. The sort of pale yellow here, this is a lipid membrane. This is a phospholipid bilayer. You should know about phospholipid bilayers. It's the same thing that makes the membrane of a cell. And then embedded in that lipid membrane are all of these complex molecules. Some of them are ATP synthase molecules. These are proteins that manufacture ATP using an electron or a proton gradient. I'll show you what I mean by that later. But we've also got these two green ones that I want to focus your attention to. PS2 and PS1. Photosystem 2 and photosystem 1. What are photosystem 2 and what are photosystem 1? Well, figuring all this out took a long time, and the way we teach photosynthesis now kind of obscures the long history of figuring out just how this works. We're going to start with photosystem 2, which was the second photosystem to be discovered and described. That's why it's called 2. But it's actually the first photosystem in the process of the light reactions. So take that dark green photosystem 2, and now think of it as a whole collection of pigment molecules surrounding a little reaction center. These pigment molecules are activated by photons of light. They're excited by photons of light. When they're excited, the electrons in these pigment molecules move around and transfer the energy of those incoming photons to the reaction center. When the reaction center is sufficiently charged by one molecule of, I think, or let's see, basically a photon with a wavelength of 680 nanometers is enough to excite this, this reaction center. Maybe don't worry about that too much. Just know that light delivers energy to these pigments, and these pigments transfer that energy to the reaction center. When the energy is transferred sufficiently, the reaction center is excited. When the reaction center is excited, there's an electron that leaves the reaction center and travels down an energy gradient along a series of molecules. So it travels along an electron transport chain from the reaction center of photosystem 2 to the reaction center of photosystem 1. This electron transport chain happens on the membrane here. So it's moving among the different molecules that are embedded in the membrane, like cytochrome B6F. So the electron's moving across these regions from photosystem 2 to a photosystem 1 over here, for example, or over here. So excited electron leaves the reaction center, travels down the electron transport chain. As it travels down the electron transport chain, it loses some energy until finally it arrives at the reaction center of photosystem 1, the first one to be discovered. Because the electron left P680, left this reaction center and traveled to this one, it needs to get an electron back. It needs to replace the one it lost. This is where water comes in. There's a, what's the word I'm looking for? There's a system in place to break water apart, to oxidize water, move that freed electron to the reaction center and release oxygen gas. And it's called the oxygen complex because it produces gaseous oxygen. But really what it's doing is it's getting an electron to replace the one that's got sent down the transport chain. So the water that's used for photosynthesis is just used to get an electron to replace this one, and the oxygen is released as a waste. Okay, let's go back to our excited electron, the one that traveled down the transport chain. It got to photosystem 1, and this electron that's now at the new reaction center is then re-excited to an even higher level here. That's what the little star means. It means it's excited. Now, I just revealed this ATP to ADP reaction here. As the electron is traveling from P680 to P700, and energy is lost because we're going down this energy state gradient a little bit, down the hill. Some of that energy that's lost is used to synthesize ATP, synthesize these energy storing molecules. And we'll see how that's used in just a moment. But once it got here, it was re-excited by a new antenna complex up here. And then the electron travels down another electron transport chain. And as it does so, the energy is stored as NADPH, a new energy storing molecule, just like ATP. So you got two different energy storing molecules that are all charged up just by using the antenna complexes to excite the electrons, send them down the chain, excite them again, and send them down the chain. It's pretty complicated, right? But it's two antenna complexes exciting electrons and then trying to use that energy to create these energy molecules. And then in the process, you got to replace the electrons with water. That's part one. That's the light reactions. The point of the light reactions is to convert that solar energy into chemical energy. Here's where the ATP and ADPH are produced, and they're sent over to the Calvin cycle. So we're going to do the Calvin cycle at the start of the next class. Make sure they know how this system works. We call this the Z scheme, because if you turn it on its side, it looks like the letter Z. Excitement, electron transport chain as the diagonal, and then excitement again. And then we got another diagonal. But the point is the diagonals are where the energy storing molecules are created. And the first reaction center is where the oxygen evolving complex is that releases oxygen and takes that electron. We also have readings associated with this in the book, and we'll pick up with the Calvin cycle next time.
Do you know what page or chapter the readings in the book are?
It should be posted on the schedule, on the daily schedule. And if you have a moment now and you want to do it fresh, it's a good idea. In that plants section, I think here. Here we go. Page 104, 105 is where you're going to want to look. Yeah, chapter 6, plant adaptations to the environment. Paul, you're muted.
Yeah, it's in the readings column in the schedule. It's 6.1 to 6.5 are the sections of the chapter.
Yeah, just the first couple parts of chapter 6.
Okay, thank you.
Sure, no problem.